The 2023 Lethbridge High School iGEM team presents Analyze This, an ANA-based biosensor for the detection and prognosis of autoimmune diseases.
What are ANAs? Anti-nuclear antibodies, also known as ANAs, are antibodies that mistake naturally occurring proteins in cell nuclei for dangerous foreign proteins and attack them. Large ANA presence can indicate an autoimmune disease caused when the immune system attacks an individual's tissues and organs. Different autoimmune diseases present specific ANAs.
Currently, most autoimmune diseases are diagnosed via antibody-based biosensors. These biosensors utilize patients' samples, usually blood, to observe the number of antibodies associated with autoimmune diseases. For example, indirect immunofluorescence investigates ANA presence in samples and is the most often used test due to its high sensitivity compared to alternative methods. Generally, ANA biosensors are more effective in diagnosing autoimmune diseases since they can detect autoimmune diseases early.
Current electrochemical biosensors give quick and accurate responses in simple procedures. This is thanks to the help of electroanalytical devices coupled with the selectivity of biorecognition molecules. This gives extremely precise readings, but electrochemical biosensors suffer from high cost, short lifetimes, and low levels of stability.
Our solution is to create a new biosensor to detect ANA. It is created with the goal of being accessible for everyone to purchase and be used in the comfort of one's home.
Analyze This will minimize patient discomfort by using saliva or urine. Analyze This will also allow for earlier detection of ANAs and hence autoimmune diseases, in turn allowing patients to seek treatment earlier and more effectively combat symptoms while reducing medical waste from testing. Our ANA biosensor design will be based on work done by Stuart Lowe's lab. In their 2022 paper, they created a switchable GFP biosensor that can change color, green to yellow, upon binding to a ligand. The protein's original N and Z termini was changed to have the N terminal start at 11 and the Z terminal at 10. A new linker region between the 11 strand and the 1 strand was added. A new 10 strand was added to 11 to create a new N terminal. This secondary 10 had tyrosine 203 instead of 309 203, which changes the fluorescence emission from green to yellow. Additionally, the amino acid linker between 11 and 10 was replaced with a disordered recognition domain, CPFKBP in blue. Upon binding to its target FK506, the CPFKBP domain undergoes a conformational change and the 10Y203 strand is then included in the beta barrel and replaces the original 10T203 strand, thus changing the emission from green to yellow.
This technology could be used to detect a variety of targets and expand it to use chromoproteins instead of fluorescent proteins. Chromoproteins and fluorescent proteins are functionally similar. Chromoproteins have an advantage for our project since no specialized equipment, such as a fluorescence spectrometer, will be required to observe the color change. Our biosensors will use TS Purple, a protein engineered by the Uppsala 2013 iGEM team, and has a chromophore sequence of CMYG. Another chromoprotein, AS Pink, has the same chromophore sequence. We created 3D models using Swiss model of TS Purple and AS Pink since no structural data was available. Both proteins have the common beta barrel shape of most fluorescent and chromoproteins with the chromophore running through the center.
By doing an amino acid sequence alignment, we could identify the residues that may contribute to the color difference. Through systematic site-directed mutagenesis, we'll identify the essential amino acids with the goal of creating a biosensor that can switch from purple to pink upon ligand binding. We also require epitope sequences to trigger the color change upon binding to ANAs. These will be identified using the available ANA binding data and analyzed for amino acid sequences that would be generally unstructured until binding to the ANA, thus causing the conformational change and the color switch of our biosensor. The engineered chromoprotein biosensor will be codon optimized for expression in E. coli and cloned into a plasmid vector. The DNA will be transformed into E. coli cells and expressed with IPTG induction. Protein expression will be completed overnight at 18 degrees Celsius. Cells will be collected, lysed, and the engineered protein will be purified. Finally, in vitro testing of the color switch will be completed by combining the engineered biosensor with ANAs. We will present our work at the 2023 iGEM Jamboree.
Before getting into University of Lethbridge's lab to work on Analyze This, team members will be mandated to complete a WMIS safety course. Though, ethical considerations such as patient confidentiality, procedural awareness, and equal access to Analyze This must also be considered.
Thank you for watching. Our team hopes Analyze This can make the diagnosing of autoimmune diseases that affect more than 320 million people much easier.